when I knew the Wilsons were coming today, I decided to bring the bulldozer so that people could see it. And I'll just take a couple of minutes here because we've had a long afternoon already today and tell you a little bit about the bulldozer. What was I talking about that day when we were in the room? You know, what are things that are happening right now that we're so excited by? Well, as I mentioned, in the last year, the last 12 months, the first gene replacement therapy ever in human beings for retinal disease occurred. And this is after 20 years of thinking about doing it. And our group is now working with these same scientists at the University of Philadelphia to begin doing the same treatment that was first done in the past year here at the University of Iowa. And I actually believe, because of the strength of the genetic unit here, that we are in a wonderful position to really be the world leader in this type of treatment, and people can come from all over the world to have those extremely rare 4 patients per year in the US kinds of things treated here. So that's a real cutting edge kind of thing. But what's the bulldozer? Well, I was telling you about the genes, and Dr. Allward told you about glaucoma genes, and Jim told you about macular degeneration genes, and lots of genes have been found, but you frequently don't think about the ones that haven't been found yet. So for example, in Project 3000, the great news is that maybe 2/3 of the genes are found, and so we can get these patients mailed in from all around and we can find them, and if we have 1,000 patients, 650 of them are found. And then what about the other 350? The other 350 people's genes haven't been found yet, and some of the samples we have in our collection are 20 years old, and we've been looking and looking and looking and looking for these things. And the genome is just an amazingly large place. It's an amazingly large place. How big is the human genome? We have 2 copies of everything except men on the X chromosome. They only have 1 copy of that. But this human genome has 3 billion units of information on each of these 2 copies. And I've learned in the years of working on these big projects that humans don't deal very well with numbers like that. Like our federal government, I think, doesn't deal very well with the, numbers like that. They can't quite tell the difference between a billion and a trillion. It rhymes with illion, so it's okay. But anyway, if one of these units of genetic information was the size of a common penny, then 1/2 of our genome would go around the Earth 1.5 times at the equator. And each one of these places in the genome has 4 possible choices. It can be an A, a T, a C, or a G. And many of the diseases that we try to diagnose with our molecular tests, we're looking for one change. Let's imagine these pennies have 4 dates: 1980, 1981, 1982, 1983. 1.5 times around the Earth, we're trying to find the one penny that has had its date changed from 1981 to 1982. That is the magnitude of the problem that we're trying to solve. So back when I was in graduate school, we would work with 1 gene in 1 person at a time with the fledgling technology of DNA sequencing. And so in my PhD thesis, I sequenced 4,000 base pairs of DNA in 14 months. 4,000, 14 months. So in the last couple of years here at the University of Iowa, we've acquired this new technology that's called pyrophosphate sequencing. And that's what the bulldozer is. So this thing is capable of sequencing 100 million base pairs of DNA in 8 hours. So I've told you what the numbers are. How does that calculate? How long would it take the instrument to do the work in my PhD thesis? How long? 1 second. Okay? So that wasn't 200 years ago. It seems to some people like it was 200 years ago. One of the people that works in my lab the other day asked me who the Beatles were. Made me feel a little old. But anyway, this is what's happening with the technology now. Each 6 or 7 years, not 6 or 7 decades, but each 6 or 7 years, something that used to take us a year takes us 2 weeks or a week. But the experiments get more expensive, the instruments get more expensive, they get way, way, way more powerful. And so for those of us that are trying to look in those patients whose genes haven't been discovered yet, having a Hubble Space Telescope type instrument that can look in millions of units of information all at once is an exceptionally exciting thing.